Welcome back to the lecture 5 of our cell series. In today's lecture, we will be learning about double membrane bound organelles and cell structure. So, firstly we have mitochondria, which is also known as the powerhouse of the cell. It is a double membrane bound organelle. It is the site of aerobic respiration. It's divided by fission. It produces cellular energy in the form of ATP, also known as adenosine triphosphate, and hence it's called the powerhouse of the cell. It has two membranes, one is the outer membrane and the other is the inner membrane, which divides its lumen into aqueous compartment and the outer compartment and the inner compartment. The inner membrane forms a number of foldings called cristae, which increases the surface area. The matrix also possesses single circular DNA, few RNA molecules, ribosomes required for the synthesis of proteins. Now let's see the structure of the mitochondria. It is the outer membrane of the mitochondria. The inner membrane has numerous foldings like structure which are known as cristae, which increases the surface area. It contains DNA, ribosomes and matrix and intermembrane space. This is all about the mitochondria. Next we have plastids. It's also a double membrane bound organelle which is found in all plant cells and in glenoids. Based on the pigment, plastids can be classified into Chloroplast, Chromoplast and Leucoplast. So firstly we have is Chloroplast. Now Chloroplast contains chlorophyll and carotenoid pigments, which are responsible for trapping light energy essential for the photosynthesis. Next we have chromoplast. These contains fat-soluble carotenoids pigments like carotene, xanthophylls, which gives yellow, orange and red color to the parts of the plant. And lastly we have leucoplast. These are colorless plastids of varied shape and sizes. So the types of leucoplast we have is amyloplast. It stores carbohydrate. We have is aleoplast. It stores oils and fat. And lastly we have aleuroplast. It stores protein. And now we're going to learn about chloroplast in detail. So it is found in the mesophyll cells of green leaves of plant. It is a double membrane bound organelle. The shape of the chloroplast varies. It can be lens, oval, spherical, discoid or ribbon shaped. Now the number of the chloroplasts also varies from 1 per cells of chlamydomonas to 20 to 40 per cells in a green alga. The inner membrane of chloroplast is less permeable. The space is limited by inner membrane of chloroplast is called as stroma. The number of organized flattened membranous sacs are called thalloids. The thalloids are arranged in stacks like piles of coins are called grana. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA along with ribosomes. Now the chlorophyll pigments are present inside the thylakoid. The ribosomes of the chloroplast are smaller, these are 70s than cytoplasmic ribosomes which are 80s. So now let's study about the structure of the chloroplast. It is a double membrane bound organelle containing outer membrane and an inner membrane. The flattened members are called thalloid and the group of thalloid coin and arranged like a piles of coin is called as granum. The luminum is called stroma and then the intermembrane space and the stroma lemma is present inside the chloroplast. Next we have other cell structures. Firstly we have a cytoskeleton. It is an elaborate network of filamentous proteinaceous structures present in the cytoplasm. It could be microtubules, actin filaments and intermediate filaments. The function of these cytoskeletons is that they provide mechanical support, motility, and maintenance to the shape of the cell. Next we have is ribosome. These were discovered by George Palade in 1955. The ribosome composed of ribonucleic acid and proteins. They do not surround by any membrane. There are two types of ribosome, 70S and 80S ribosome. Now in 80S and 70S ribosomes, S denotes the Sedberg unit, which stand for the sedimentation coefficient. Of ribosome in detail. So the 70S ribosomes are found in prokaryotes. They are made up of two subunits, larger subunit and smaller subunit. Now the largest subunit of 70S ribosome is 50S and smaller subunit is 30S. Now 80S ribosomes are found in Now the larger unit of 80S ribosome is 60S and smaller unit is 40S. This is all about the ribosome. At last we have cilia and flagella. Now cilia and flagella are seen both in eukaryotic and prokaryotic cell, but eukaryotic flagella different from the prokaryotic flagella in terms of its structure. Now it is a hair-like outgrowth of the cell membrane. So let's study about cilia first. These are the small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Now flagella. These are longer and responsible for the cell movement. They are covered with the plasma membrane. Their core is called as axoneme, possesses number of microtubes running parallel to the long axis. 9 doublets of radially arranged peripheral microtubes. These are 9 doublets of peripheral tubules which are connected to each doublet with the interdoublet bridge, which run parallel to the axoneme. And in center there are central microtubules. Now the central microtubes are connected by bridges and also enclosed by a sheet, which is known as central sheet. It has 9 radial spokes. These are radial spokes. So this is all about today's lecture.